reversible reactions represent the steps in what's called an enzyme catalyzed reaction. Okay. So what you have first is you form what's called an enzyme substrate complex, where the enzyme and the substrate bond. The substrate is the material that were before the reaction. Okay, it's your reactants. The enzyme substrate complex is a transitional state. So it comes together, they bond, then they separate. When they separate, that's when the product is formed. Okay. Enzyme product is the enzyme product complex. So you have the substrate combined to the enzyme. Okay. And the substrate is converted to the product, then the product is released. So these are the steps involved. You have the enzyme plus the substrate come together to form the enzyme substrate complex. Okay. This is a transition state. Okay. Then it forms the enzyme product complex. Then the product separates from the enzyme, regenerating the enzyme. So there's your enzyme and there's your enzyme. Remember the definition of an enzyme? It's a catalyst. Okay. So it has to be regenerated and not used up in the reaction. Okay. And those are the steps involved in the enzyme substrate complex. Okay. And all those are reversible. So it can go, you can go back this way and then release the substrate back again. Every step's reversible. It all occurs by equilibrium. All right. So the part of the enzyme combining with the substrate is what's called the active site. That's where the substrate binds to the enzyme. Okay. We have different characteristics of this active site. Okay. It's in the pocket or a cleft where the substrate can fit into. Okay. There's R groups at the active site. Okay. Those are called your catalytic groups. Those can form hydrogen bonds. Those can form disulfide bonds. They can form salt bridges. They can form van der Waals forces. They can form other forces that help hold that substrate into the enzyme from the R groups that are inside the active site. Okay. The shape of the active site, okay, is complementary to the shape of the substrate. So if the shape of the active site is like that, then the substrate is going to have the matching shape so it can come in here and bind with the appropriate types of bonds. Okay. You're not going to see an enzyme that looks like that and a substrate that looks like that. I mean, that's not going to fit in. Okay. So the shape is going to be complementary to the shape of the substrate, the shape of the active site. Okay. So the enzyme attracts and holds the enzyme using weak. This should say substrate. That didn't make sense. I have to fix that on the slide. So the enzyme attracts and holds the substrate using weak non-covalent interactions because they have to be separated again. You're not going to covalently bond it to it. Okay, it's going to be reversible bond. It's going to form the bond, a weak non-covalent interactions that hold the substrate to the enzyme at the active site. Like I said, hydrogen bonding, salt bridges, hydrophobic interactions, disulfide bonds, things like that. Just like we saw with our primary, secondary, and tertiary shapes of proteins, which makes their shape. So conformation of the active site determines the specificity of the enzyme. Okay. The type of the active site determines how specific is going to be the substrate that binds to that enzyme. Okay, I think this is right where we left off right here on last time. This is your lock and key model. It's just like the front door of your house, like a lock and key that fits. The enzyme is assumed to be the lock. So your enzyme is the lock, substrate is the key that fits into it. Okay. Enzyme and substrate are made to fit exactly. Okay, this model fails to take into account protein conformational changes to accommodate a substrate molecule. So different molecules can fit into different enzymes sometimes, so it doesn't really take that into consideration. So it's just one of the models we propose for it. And you can see the enzyme and the substrate, okay, and the enzyme substrate complex. You can see this is like a lock and key. This is going to fit right into there just like a lock and key. And then it binds right to it. Okay. You can also have what's called an induced fit model. Okay? The induced fit model of enzyme action assumes that the enzyme active site is more a flexible pocket whose conformation changes to accommodate the substrate molecule. That seems to fit most a lot of the research that we've done. Okay. So here's the enzyme right here. Okay, and you see it's similar in shape to the substrate, but as the substrate moves in, it changes its shape to match it exactly. It conforms to the shape of the substrate. That's an induced fit model. When the substrate approaches the active site modifies itself, changes its conformation to match the substrate exactly. It induces it to change. That's what's called an induced fit enzyme model. Okay. So the overall shape of a protein is maintained by the weak interactions, like we said, the hydrogen bonds, disulfide bridges, salt bridges, and hydrophobic interactions. Okay, those are the same shapes that are used to determine our tertiary structures or proteins, same things. Okay. So at any time, a few of these weak interactions may be broken by heat or local change in pH. You can break those bonds that are holding that enzyme substrate complex together. Okay. If only a few bonds are broken, they'll reform very quickly. Okay. The result is that there's a brief change in the shape of the enzyme as the substrate approaches it and induces it to change. Okay. So the protein or enzyme can be viewed as flexible molecule, okay, changing shape slightly in response to minor local changes. Okay. So the enzyme will change shape to match the substrate exactly in the induced fit model. So we're going to compare the lock and key model and the induced fit models of enzyme substrate complex and their binding. Okay. So the lock and key model. Okay, so you have the first one is the lock and key model of the enzyme substrate complex. Binding assumes that the enzyme active site has a rigid structure, okay, and it's precisely complementary shape and charge distribution to the substrate. So the substrate fits in exactly as it is shaped. That's the lock and key model. Only one key fits one lock. The induced fit model, okay, of enzyme substrate complex assumes, okay, there we go, assumes that the enzyme binds to the substrate. The shape of the active sites conforms precisely to the shape of the substrate. Okay. So your active site conforms precisely, but it's induced, it changes. When the enzyme binds to the substrate, the shape of the active site changes to match the shape of the substrate. So the shape of the substrate may also change when it bonds into the induced fit model. Okay. But they change to modify one or the other to make an induced exact fit. Whereas lock and key, they're rigid. They're not going to change. Induced fit means they can induce one another to change to match and form that enzyme substrate complex. The induced fit model is more accurate. We know that proteins are flexible molecules. We've seen by their structures, by how they bend and how they shape, and the bonds that hold them together. What is the relationship between an enzyme active site and its substrate? Okay. The active site is a part of the enzyme where the substrate binds to. They have to be complementary to each other. Okay. So the enzyme active sites are pockets or clefts in the surface of the enzyme. Okay. The shape of the active site is complementary to the shape of the substrate. The substrate fits neatly into the active site of the enzyme. They match and they bond together. And they're held together by weak bonds. They're not covalent bonds. Okay, so the enzyme attracts and holds its substrate by weak, non-covalent interactions. Okay, just like the tertiary structure of a protein. Same types of bonds. Okay. The conformation of the active site determines the specificity of the enzyme. Okay, so it's only going to match that substrate that fits into the active site well. Okay, and that's the only way that can be used in the reaction. It has to fit into the active site so it can be used. Okay. So you have to match the active site, the shape of it, so you can induce it to fit the substrate in. Okay. If it's too far off, enzyme's not going to be specific enough to match into that enzyme. So specificity, we're talking about how they match up to each other. So for enzymes and substrates to react, the surface of each must be complementary. They have to complement one another. There's got to be a fit in there. So enzyme specificity, by definition, is the ability of an enzyme to bind only one or very few substrates. Okay, so very few, but have to have the same shape, thereby catalyzing only a single reaction. Okay, it's only going to bind things that have very specific shape. Okay, compare the following two reactions. Okay, you got urea and methylurea. Urease is the enzyme. This will bond, the shape of this will bond with urease. They'll bond together and release carbon dioxide and ammonia and break down urea. Okay. Methylurea, it has a CH3 group on, the only difference is this CH3 group on the end. Okay, that changes the shape and it will not bind to the urease. It will not bind to the urease, so they can't come in contact. They can't form the enzyme substrate complex, so no reaction occurs. Okay. So urease is very specific or has a high degree of specificity. Okay. So urease bonds to a very specific shape. And if it's off in that area, if off in that shape, it's not going to bind. Okay, that's specificity, how it's going to bind to it. Okay. Classes of enzyme specificity. You have different ways of classifying or categorizing the way these enzymes bind. Absolute means the enzyme reacts with only one substrate. That's it. It's very, very specific. That matches more like your lock and key model. It's one exact fit. That's what absolute classification is. A group classification. Enzyme catalyzed reactions involved in any molecules with the same functional group. So anything with a alcohol function, OH, it's going to bind on that OH group. Anything with a carboxyl group, COOH, it's going to bind a carboxylic acid group. Okay. Ketone, C double bond O. Okay. Anything that's going to bond to this specific group, to the functional group. So anyone that any enzyme that catalyzes reaction with any functional group is called a group classification. So you have absolute and you got group. And you got what's called a linkage. Linkage is where the enzyme catalyzes the formation or breakup of only a certain category or type of bond. So type of bond, carbon nitrogen bond, carbon hydrogen bond, carbon carbon bond, a double bond, a specific type of bond is what catalyzes. This is a linkage classification. And they're stereochemical. So you have four types. You have absolute, group, linkage, and stereochemical. In stereochemical, enzyme recognizes only one of two enantiomers. Remember, enantiomers are the rotation, which rotates plane polarized light, either right or left. So D or L. They have like D or L sugars. Okay. D or L amino acids. Okay. It's going to catalyze only one of those two. The other enantiomer will not fit into the bond. That's a stereochemical active site. That's the classification of that enzyme. It'll only catalyze either the D or the L shape, not both. So remember, they're non-superimposable. They're not going to line up the same way. Okay. We only use D sugars and L amino acids for our bodies. Okay. All our bodies use our D sugars, and because of amino acid, we use the L form of the amino acids. And we on the enzymes only recognize that particular stereoisomer of each one. It'll only recognize the D sugars. It'll only recognize the L amino acids. Okay. Transition states and product formation. Remember, we have the transitional state. The transitional state is when the enzyme and stuff come together, and now things can happen. It's holding them in close proximity, so it modifies it so enough so a reaction can occur. It lowers the activation energy. It brings things together usually. So how does this enzyme make a faster chemical reaction? How does it do it? Normally in the lab, you just heat things up, makes it go faster, but you can't do that in the body. So as a substrate, that's what's coming into the enzyme, interacts with the enzyme, its shape changes. Okay, this new shape, okay, has a less energetically stable. Okay, so it's going to break apart easier now. It's going to change. The new shape that's induced, that forms from the substrate, makes it easier to break those bonds and move things around to form the products, because it's in a less energetically stable state. Okay, this transition state has features of both substrate and product, and falls apart to yield the product which dissociates from the enzyme. So you produce your enzyme back in the original form, which is like a catalyst, Cassetti catalyst. Remember, it has to regenerate itself. Okay, and then it releases. The substrate comes in. The bonds are changed. The shape is changed. So now it can react. It can change. It can modify itself, and then that product is released once it's modified itself. Okay, so again, this is the same thing we started last time. Your enzyme and your substrate are going to come together. They're going to form an enzyme substrate complex. Okay, this is a transitional state. Okay, this can go either way. It goes back to the original, or it comes out. This is not a long-lived state. It's transitional. What happens is it makes the substrate change. Okay, so the substrate is now changed into the product, and it's bound to the enzyme. Then that enzyme releases the product and regenerates your enzyme. So you start with this enzyme and you end up with this enzyme. You start with a substrate, you end up with a product. And those are just the steps in between. Okay. Enzyme and substrate bind, substrate starts to bend and change, goes into the product, then releases itself from the enzyme. That's basically how it works. Okay, there are different types of transitional state changes. Okay, there's different ways to make that substrate change its shape so it's less energetically favored. So the enzyme might put stress on a bond, making it easier to break that bond. Okay, it may bind to the enzyme, and it binds in such a way that it twists itself and puts stress on one of the bonds, making that bond easier to break to form the product. Okay, and there's an example of it. So here's your enzyme and substrate. They come together. This is your enzyme substrate complex. Okay, and you can see this piece here gets modified and it starts to stick out and it bends this bond right here. And as it bends the bonds, it makes that bond easier to break. So then the bond does break. This is your enzyme product complex. Okay? And then the product is released, and now you have your product. And you're regenerating this enzyme just like it is when it started. Okay? Because you have to regenerate. It's a catalyst. It has to be regenerated at the end. Can't be used up in the reaction. Okay. Okay, or the enzyme might bring two reactions into close proximity and give them a proper orientation so that they can bind. So they can approach one another and form a bond. Okay, this is an example of glucose and fructose. Okay, the glucose and fructose combine to form sucrose. Okay, by the peptide linkage there. Okay, so you have your enzyme. This is number one. You have your enzyme here, and this is your substrate. The substrate combines and forms into it. It brings these so the orientation is correct, so this 1,4 linkage can form. Okay, and then it forms. Remember, dehydration reaction. We lose an OH and we lose an H, and there's your water that's given off. And now these two come together and form. This is your enzyme product. This is your enzyme substrate complex. This is your enzyme product complex, and you release the product, regenerating your enzyme. Okay. Okay, so you got your enzyme substrate complex. It comes in and it positions it so they can match and form that 1,4 linkage. Okay. And then the bond gets formed here. So you got your enzyme product complex. Water is released, dehydration reaction, and you form your product gets released and you reform your enzyme again. Okay. Another way is the enzyme might modify the pH of the microenvironment, donating or accepting a hydrogen. Okay. If it becomes acidic, it's going to donate a proton because you have H pluses. If it becomes basic, it's going to accept a proton. Right. So summarize 3 ways in which an enzyme might lower the energy of activation of a reaction. We talked about 3 ways. An enzyme might distort a bond, allowing it to break easy. An enzyme could bring 2 reactants into close proximity. So they're in proper orientation for the reaction to occur, and they can form a bond. So you can distort a bond, making it break. You can bring them into close proximity and hold them in the right order so they can form that new bond easier. Okay, or the enzyme could alter the pH of the microenvironment. So it could either accept a proton or donate a proton to change the substrate. So those are 3 ways. Okay, then an enzyme might lower the activation of a reaction. It lowers the activation energy, so the reaction can occur quicker. That's all it does. It lowers the activation energy. What is the transition state in an enzyme catalyzed reaction? The transition state is the enzyme substrate complex as it changes into the enzyme product complex. Okay. The transitional state is when the substrate is bound to the enzyme. Okay. Remember, you have your enzyme plus your substrate, forms an enzyme substrate, then forms an enzyme product, then it releases the enzyme plus the product. So this is your transition state. The enzyme substrate is transition state. The enzyme product is a transitional state. It's a state that's formed as another one is created. All right, HIV protease inhibitors and pharmaceutical drug company design. This is one of the drugs combined for and during HIV treatments that we actually started using. And it started back in the 1980s. Okay, in 1981, the Center for Disease Control in Atlanta recognized a new disease syndrome called AIDS, acquired immune deficiency syndrome. Okay, this was back in 1981. Right. The syndrome is characterized by an impaired immune system, which we know, a variety of opportunistic infections and cancers, brain damage that results in dementia. Okay. It attacks your brain, it forms different types of cancers. What you see in the mouth is we call Kaposi sarcoma, which are black, purplish bumps in the mouth that form. That's one of the almost indications of AIDS. Okay. The AIDS virus decreases your immune system so it can't suppress different types of things, and you get infections and cancers, brain damage, etc. from that. Okay. It soon became apparent that the disease is being transmitted by blood and blood products, as well as by sexual conduct. Okay. That's how they're transmitting the AIDS virus. Okay. The earliest drugs that proved effective in the treatment of HIV infections all inhibited replication of genetic material of the virus. Remember, all a virus is genetic material. And it lacks the ability to replicate itself. A virus has to infect a host cell, human cell in this case, okay, to be able to replicate. It tricks over the nucleus and it uses the cell's machinery to replicate itself. Okay, that's how the virus works. Viruses can't replicate themselves without being infected into a regular cell. While these treatments were initially effective, prolonging the lives of many, it was not long before viral mutants resistant to these drugs begin to appear. Because remember, viruses mutate at an extremely fast rate because their generations are so quick. You know, they replicate every 20 minutes versus every 20 years with humans. Okay, so they can, as they evolve, they change, become resistant to the different types of drugs that we use for that. Okay. So a new approach was needed. So the enzyme that's necessary for viral replication, because this virus has an unusual strategy for making all of its proteins. Okay. So the virus uses a specific enzyme to replicate its proteins. Okay. Rather than make each protein individually, it makes a large polyprotein. Okay. That's got to be cut by the HIV protease to form the final proteins required for viral replication. So it makes its own enzyme. It forms this long protein, okay, and then cuts it into pieces using what's called protease, and it forms the final product so the virus can replicate. And that's what it looks like. This is a three-dimensional, the HIV protease. Okay. This is the tertiary configuration where it's shaped around and all the bends and everything are inside the molecule. That's the active form. And you see some complex areas there as it folds and makes a tertiary structure. Since scientists realized that the enzyme was essential for HIV replication, they decided to engineer a substance that would inhibit the enzyme by binding irreversibly to the active sites. If you bind irreversibly, that means it's not going to come off. And we're going to bind to that active site. Okay. If you bind that active site, it can't cut its enzyme, so it can't become functional. Basically, the HIV virus cannot replicate if you block that active site on the enzyme. Okay. The challenge was to design a molecule that would be that plug, that would form it and have that shape and bind to that HIV replicase. Researchers knew that the primary structure, amino acid sequence of the HIV protease from the earlier nucleic acid sequencing studies. Okay. So by 1989, they also had a very complete picture of the three-dimensional nature of the molecule, which they obtained by X-ray crystallography. So putting all this together, okay, into a computer modeling program, they could look at the protease from every angle. They could move around, they could determine what the active site was. They could see locations of the R groups of each amino acid in the active site. Okay. This kind of information allowed the scientists to design molecules that would be complementary to the shape, okay, and charge distribution of the enzyme's active site. So structural analog of the normal protease substrate. So it took the protease, they found the active site, they mapped out the shape that it's going to be, okay, because they knew the R groups, they knew the amino acid sequence, so they mapped out the shape, okay, and now they could find something that's going to complement into that shape exactly. So it was not long before the scientists produced several candidates for the HIV protease inhibitor. Again, so if you inhibit or block HIV protease, which is the enzyme the virus uses for replication to trigger the cell to replicate itself, you can stop HIV from replicating. That's the theory. The many trusted drug candidates patchwork being introduced in the market is safe and effective. So scientists had to show the candidate drugs would bind effectively to the HIV protease and block its function. That would inhibit the virus from replicating in the body. Properties like solubility, the efficacy of absorption by the body, the period of activity in the body, and the toxicity of the drug candidates all had to be determined. It does you no good if the drug you're introducing is toxic to the body and it kills it without the AIDS virus. You got to make sure everything matches and everything can be compatible with the human body first. So by 1996, there were 3 protein inhibitors available to combat the HIV infection. Okay, there are currently 7 of these drugs in the market today. Okay. So in many cases, development testing of a drug candidate can take up to 15 years. It's not a fast process because there's a lot of things you have to check for. In the case of the first HIV protease inhibitors, the first 3 drugs are on the market in less than 8 years after the discovery of HIV. Okay. This is accessible to the urgent need for HIV treatments and the technology that's available. Cofactors and coenzymes. Enzymes usually work with other vitamins or other factors in the body to produce their effects. Okay. An active enzyme is called a holoenzyme. That's your active enzyme. Because a lot of enzymes aren't produced as being active until they get to this point. That's supposed to have their effect. So the active enzyme is called the holoenzyme. The polypeptide portion of the enzyme, which is your amino acid chain, is called an apoenzyme. Okay. A non-protein prosthetic group that's on it is called a cofactor. That's what binds to the enzyme. It helps it accomplish or activates that enzyme, accomplish its effect. So cofactors are bound to the enzyme for it to maintain the correct configuration of the active site. So the enzyme's made, okay, it's an apoenzyme, it goes around, travels, and it goes to the point where it needs to become active. It binds with a cofactor. This cofactor then changes the shape of the active site, so now the enzyme is active. That's what cofactors can do. Okay, cofactors can be organometallic compounds. Okay, they can be metal ions. They can be organic compounds. Okay? Any of those three. So here we have your apoenzyme. This is the enzyme, which was your tertiary structure. And the active site looks like that. Okay. This is the enzyme. This is your substrate. Is it going to fit? No, it's not going to match. Okay. When copper is released in the solution by it, the copper 2+ ion, it binds to the enzyme. When it binds to the enzyme, it changes the shape of the active site. So now you got a cofactor and enzyme complex. You got it changing the active site. So now the enzyme is active. Now the substrate can bond to the enzyme. Okay. So you have an apoenzyme, which is just made from your amino acid chain, tertiary structure. Okay. The substrate doesn't fit. Okay. No enzyme substrate complex, no reaction. Okay. Copper is introduced as a cofactor. The cofactor binds to the enzyme. Now it is not an apoenzyme anymore. It's a holoenzyme. It's an enzyme that is functional. The active site, basically from back there, is active. Okay. The substrate can bind to it, and it can form the enzyme substrate complex. And once it binds, then the reaction can occur by one of the three processes that we talked about, modifying the bond, adding it together, or so on, or changing the pH of the reaction. Okay. So these coenzymes are required by some enzymes. Okay. An organic molecule bound to the enzyme by weak interactions or hydrogen bonds. Okay. Those are coenzymes. They don't bind covalently. The organic molecule that binds to the enzyme by your weak interactions, hydrogen bonds, salt bridges, things like that. Okay. Most coenzymes carry electrons or small groups that modify the shape. Many have modified vitamins in their structure. Okay. A lot of times we use vitamins. Vitamins become modified and they become coenzymes to make enzymes work, certain enzymes. Okay. So if you look at this, this is your, where we at? This is our enzyme. Okay. And this is your coenzyme right here. Okay. So an enzyme with a coenzyme positions to react with two different substrates, substrate 1 and substrate 2. Okay. So there's a functional group on substrate 1. The coenzyme allows the functional group to transfer from substrate 1 to substrate 2. Okay. The coenzyme sits in that active site, and it takes the group, whether it's a hydroxyl group, carboxyl, whatever it is, and transfers it to substrate number 2. Okay. So you removed a group from substrate number 1, and this becomes your product number 1, because now this has been removed from it, that little area. Okay, so the coenzyme has to be there for the enzyme to be active, and the activity allows substrate 1 to transfer a group, some functional group to substrate 2, and then substrate 1 now becomes the product because the group is gone, the group has been transferred. Okay? And the product is released from the enzyme, and the enzyme is regenerated just as it was. and it would not function unless this coenzyme was present. It allowed the transfer. So often coenzymes contain modified vitamins, like we said, as part of their structure. A vitamin is an organic substance that's required in the diet in only small amounts. That's what a vitamin is. All right. Of the water-soluble vitamins, only vitamin C has not been associated with a coenzyme. So all the water-soluble vitamins are associated with a coenzyme, except we just haven't found the one that vitamin C does yet. Okay. So here's the different water-soluble vitamins: B1, B2, B3, B6, B12, folic acid, pantothenic acid, biotin, ascorbic acid. Ascorbic acid is the only one that's unknown as to where it functions as a coenzyme. The other ones all have coenzymes which act to, decarboxylation, carry hydrogen atoms, hydride ions, okay, carriers of amino acid and carboxyl groups, okay, as that function as a coenzyme. So like we said, vitamin C, ascorbic acid, is the only one we don't know where the coenzyme fits on that into any other enzyme. So nicotinamide adenine dinucleotide, NAD, we're going to learn a lot about that as we learn about the electron transport chain and generation of ATP. So NAD is derived from niacin, which is vitamin B3. Okay, this is NAD plus here. Okay, and you can see you've added this. What happens is with the hydride ion, hydride is H minus. Okay, you've added a hydrogen to that carbon right there. There's one carbon here, and now there's two carbons. There's one hydrogen at that carbon. Now there's two hydrogens at that carbon. So what you've done is you take an NAD plus, okay, and formed NADH, which is a reduced form. So this is a reduction reaction. Okay, remember, reduction is getting rid of oxygen or adding hydrogen. Oxidation was adding oxygen or getting rid of hydrogen. So this is adding hydrogen to form NADH. This is a reduced form. And we use that in the Krebs cycle, use that in other cycles in the body to produce ATP. Okay, so NAD, nicotinamide, is derived from niacin. Okay. And that's what it looks like. You see the phosphate groups that connect the two purines. Okay, so NAD plus to NADH. Okay, you're reducing it. You're adding hydrogen to NAD. So the nicotinamide part of NAD plus accepts a hydride ion. Remember, hydride is H minus. So H plus 2 electrons gives you that negative charge. And it accepts it from an alcohol to be reduced. Okay. So the alcohol loses a proton to the solvent to be oxidized. Remember, if something's being reduced, something else has to be oxidized. If something being oxidized, something else has to be reduced. They have to occur together. Can't just pull electrons out of the air. They got to come from somewhere. Okay. So it takes the hydrogen from this alcohol and reduce it here. So hydrogen is, the alcohol is oxidized because the alcohol goes from that here to a carbonyl group. So you added more bonds to oxygen. Okay. So the alcohol has been oxidized and NAD plus has been reduced to NADH. Okay. So this is your oxidized form. Let's do this right here. This is your oxidized form. And this is the reduced form. Right? It's had a hydrogen added to it. And that's how we take NAD plus to NADH. Okay. Two other adenine dinucleotide coenzymes. Okay. NADP plus, okay, can form flavin adenine dinucleotide, FAD. You can see where there's been nitrogens added to it. and it's changed the structure on it. Okay. You've gone from this phosphate group here, okay, gotten rid of that phosphate group, and you've added the chains up here, 2 ring structures with nitrogen. Okay. There's two different things we're going to use to oxidize and reduce in the ATP chain. So we have NAD plus and we have FAD, which we'll use. And because you're going to add a hydrogen to these nitrogens right here and add another hydrogen right in there when we use those to reduce them. Okay. So why does the body require water-soluble vitamins? A lot of the body's water. Okay. Water-soluble vitamins are required by the body for the synthesis of coenzymes that are required for the function of a variety of enzymes. Okay. So for enzymes to become functional, to be active, the active site requires a coenzyme. Now, a lot of the coenzymes are derived from our water-soluble vitamins. Like I said, we got all of them except for vitamin C that we know exactly where they go. Okay. So what are the coenzymes formed from each of the following vitamins? What are the functions of each of the coenzymes? Again, you look at that chart that we went through before, you'll be able to tell exactly what those are. Pantothenic acid. Pantothenic acid is used in the formation of coenzyme A. Okay. It functions in an acyl group transfer. So a carbon group, it transfers carbon from one substrate to another. That's the coenzyme that helps achieve through the enzymes. Niacin. Okay. Niacin is used in the formation of coenzyme nicotine adenine dinucleotide, NAD+. Okay. And NADP+, and it functions as a carrier for the hydride ion. It transfers hydrogen to those to form NADH and NADPH. Okay. So that's what niacin does. So it's the coenzyme for NAD+ and NADP+. Okay, and is a carrier for hydride. It allows a transfer of hydrogen from an alcohol back to the NAD+ or NADP+. Okay, so it causes reduction of those by adding hydrogen. Riboflavin. Riboflavin is a B vitamin. It's used in the formation of the coenzyme flavin mononucleotide, FMN, and flavin adenine dinucleotide, FAD. Okay, it functions as a carrier of hydrogen atoms. It allows hydrogen to be transferred so something can be oxidized, something can be reduced. If it's carrying hydrogen to another structure, that structure or that substrate is being reduced because you're adding hydrogen to it. Yeah. All right, and again, environmental effects on enzymes too, which you have talked about a little bit. We talked about pH, we talked about heat, denaturing enzymes, etc. So the environmental surrounding an enzyme can have a direct effect on the enzyme's function. Okay. Enzyme works best at a particular range of pH. Human body is 7.3 to 7.4. That's the range where our enzymes are designed to work, in that particular pH range. Once it exceeds that range or goes below that range, then things start to act differently. Okay. Extreme pH changes will denature the enzyme, destroying its catalytic ability. It won't let the enzyme function anymore. It'll change the shape of the active site. Okay, because if you add hydrogen, like if it gets too acidic, you're going to add hydrogen, and it will change the shape of that active site. If it's too basic, okay, you'll take away hydrogen. Again, change the shape of the active site. And that will what's called denature the enzyme with extreme pH changes. Pepsin is an enzyme that's found in the stomach. Okay. Chymotrypsin is found in the small intestine. They have different optimal pHs. They're designed to function at different pHs. If you're in the bloodstream, the pH of the body is 7.3 to 7.4. If you're in the stomach, pH is 2. Your stomach's a lot more acidic than your bloodstream. Okay, extremely acidic. You have to function at a different pH. So most enzymes are active only within a very narrow pH range. Okay. The cytoplasmic cell has a pH of 7.3, 7.4. Okay, so it's mostly neutral. Most enzymes function best at that pH. But like we said, a plot of reaction versus pH for a typical reaction is shown. So if the pH is optimized, okay, velocity is how fast the reaction occurs. At 5, it doesn't occur hardly at all. Comes up here at 7. This is the peak performance. That's where catalyst is going to occur. Catalyzation is going to occur quickly, and then back it becomes more basic. Okay, it becomes too basic, too acidic. It changes the active site. The enzyme doesn't function. And most enzymes are designed to work around pH 7, which is what our body is. The pH at which the enzyme functions ultimately is called the optimal pH, or the pH optimum. That's the pH where the enzyme is going to function at its optimal level. Okay, making the solution more basic or acidic decreases the rate of the reaction. It's not going to work as well if your body becomes more acidic or more basic. These pH changes alter the degree of ionization of amino acids on the R groups in the protein. Okay, once they do that for the R groups, they modify the shape of the active site. They also modify where you can have hydrogen bonding, because you're either adding hydrogen, taking hydrogen away if it's too acidic or too basic. Okay. This causes the enzyme to lose its biological active configuration and therefore become denatured. Denatured means the enzyme is no longer functional. Okay. Some enzymes must function at pH far from 7. Like we said, we're talking about pepsin in the stomach. Okay. The pH of stomach acid is about 2. Okay. 2 is a long way off from 7. Okay. As a result of secretion of hydrochloric acid by cells that line the stomach, they secrete hydrochloric acid directly into your stomach. Okay. So at pH of 2, your enzymes are not going to work unless they're specially designed to work at that pH, which some are. Okay. The proteolytic digestive enzyme pepsin, okay, must effectively degrade proteins at the pH of 2. Okay, so pepsin is functional. Pepsin works. The active site remains functional at pH of 2, not the pH of 7 like other enzymes, because of where it is. In the case of pepsin, the enzyme has evolved an amino acid sequence that can maintain a stable tertiary structure at pH 2, and is most active in the hydrolysis of peptides that have been denatured by a very low pH. Okay, low pH is starting to, you eat protein in your diet, protein goes into the stomach. The stomach has a pH of 2, that is going to start to hydrolyze the protein and start breaking it apart. As it breaks it apart, it goes into smaller pieces, and then the pepsin attacks the smaller pieces and further breaks down into smaller amino acids. Okay. So pepsin has a pH optimum of 2. Most enzymes have a pH optimum of around 7. The pepsin is designed specifically to work in the stomach under extreme acidic conditions. Okay. Just like pH can have an effect, temperature can have an effect. So an enzyme has an optimum temperature associated with maximum function. It has an optimum pH, it's also got an optimum temperature. Okay, so the rate of an uncatalyzed reaction will increase proportionally with temperature increase. So this is an uncatalyzed reaction. Okay, this is your uncatalyzed reaction. As the temperature increases, the rate increases. Okay, it's linear. That's what we do in the laboratory. That's what you do when you want to make Kool-Aid. Okay, if you take cold water, well, let's make Jello. You take cold water, you pour the Jello in. Nothing's going to happen, right? It just kind of sits there in clumps. Take that water, heat it, and boil it. That stuff dissolves right away. Okay, you increase the rate of the reaction with the increase in temperature. Okay, and the more you increase the temperature, the faster the reaction goes, the faster the little molecules are moving. Now, that doesn't work with catalyzed reactions, with a catalyst. Optimum temperature is usually close to the temperature at which the enzyme typically exists. So this is a catalyzed reaction right here, which is 37 degrees centigrade for human, 98.6 degrees Fahrenheit. Okay, so this is your reaction. As the temperature increases, Okay, it increases and it goes up to a rate, and this is an optimal amount. The rate's gonna be right here. And then as it further increases, it starts to go down. It starts to denature the enzyme. Okay, so it only goes up to a certain point. This is your optimal temperature. Just again, optimal pH, you have an optimal temperature here. Okay, the optimal temperature for most enzymes is 37 degrees for most humans. Okay, excessive heat can denature protein, making it completely non-functional. Okay, at 70 degrees, your enzyme is non-functional. At 70 degrees, if it's an uncatalyzed reaction, it's keep on going. It keeps on trucking. Okay, but enzymes have an optimal pH, they have an optimal temperature range at which they're going to function. Okay. Because heating enzymes and other proteins destroys the three-dimensional structure, their activity, a cell cannot survive very high temperatures. Okay, because the machinery breaks down. Okay, the proteins inside start to break apart. The three-dimensional, remember the three-dimensional shape, the tertiary structure. The tertiary structure is not held together by covalent bonds. It's held together by weak forces, hydrogen bonds, salt bridges, disulfide bonds, hydrophobic interactions pockets. Okay, those can all be broken by high temperatures, and they are. And once you break the bonds holding the shape together, it no more has that shape. So it's non-functional. You destroy the enzyme. Okay, so heat is an effective means of sterilizing medical instruments because it destroys the cells, bacterial cells, viral cells. Okay, it's an effective means of sterilizing medical instruments and solutions for transfer or for clinical tests, for transfusion or clinical tests. Although instruments can be sterilized by dry heat at 160 degrees centigrade, that's what kills bacteria and viruses, applied for at least 2 hours in a dry oven. Now, most offices, including my office, my dental office, I don't have 2 hours to sterilize my instruments. I don't. It's going to have much faster turnaround time than that. So we use what's called an autoclave. We have that heat, but now we add pressure to it. Now it works a lot faster. It reaches temperature, it destroys them, and it's more reliable. Okay. Autoclave works on the principle of pressure cooker. Okay. Air is pumped out of the chamber. Steam under pressure is pumped into the chamber until a pressure of 2 atmospheres is achieved. Okay. The pressure causes the temperature of the steam, which should be about 100 degrees centigrade at atmospheric pressure, to be 121 degrees centigrade at that increased pressure at the 2 atmospheres. Okay. Everybody remember from chemistry, PV equals nRT, the old gas law? If you increase pressure, I'm going to increase temperature. If I hold number of moles constant, R is always constant, and the volume constant in an autoclave. So that's just PV equals nRT. If pressure increases, the temperature is going to increase. So if I get up to 100 degrees centigrade, I crank up the pressure. Now it's at 121 degrees centigrade. So it can be a lot higher temperature, so I don't have to hold it quite as long at that temperature. And most autoclaves run around 15 to 20 minutes to sterilize a load of instruments, depending on the size and how much you put in and stuff like that. And all the autoclaves are tested weekly by I think it's federal law actually. You have to run it, but you have to throw a test strip in your autoclave and you run it with your instruments once a week and send it to a government lab, and they have to check to make sure everything's dying in there. And they put inside these test strips hepatitis B spores. Those are the hardest thing to kill. Hepatitis B spores are extremely difficult to kill. If you've killed those, you've killed anything else. I mean, AIDS virus, you can kill that with soap and water. Okay, that's easy to kill. But the hepatitis B spore is extremely difficult to kill. So that's what they put inside our test strips. So if we're killing that, we got it covered. And every autoclave has to be tested by the government every week. I send in a strip. Okay, within 20 minutes, that's like that. My autoclave runs at 20 minutes. All the bacteria and viruses are killed, and that's what sterilizes your instruments. That's how it works. This is the most effective means of destroying the very heat-resistant endospores that are formed by many bacteria of clinical interest, like hepatitis B spores. Okay, these bacteria include the genre Bacillus and Clostridium, which are responsible for such unpleasant and deadly disease like anthrax, gas gangrene, tetanus, botulism, food poisoning. All those are no fun to get. That's why instruments are sterilized. Okay. So how does a decrease in pH alter the activity in the enzyme? If you decrease the pH, you're making it more acidic. You're going from 7 to 6 to 5 to 4. In other words, you're decreasing pH, you are increasing the H plus, increasing the acidity. So what's going to do the activity of the enzyme? It's going to denature, right? It's going to make it less work, because if you're putting H plus there, that's going to change the active bindings, the active site, because the active site is held together by, say, hydrogen bonds. Now you're putting a hydrogen on there, and you're getting rid of the hydrogen bond. So the active site shape is going to change. Okay? So a decrease in pH will change the degree of ionization, it'll increase the H plus, and the R groups within a peptide. So the R groups are going to change because you put H's on those now, and they're not going to have the same bonding abilities as they did before. Okay? This disturbs the weak interactions that maintain the structure of an enzyme, which may denature the enzyme, make the active site not able to bind to the substrate. Okay. Less drastic alterations in the change of R groups in the active site of the enzyme can inhibit enzyme substrate binding. So even a small amount of change in pH can inhibit this, and that if necessary modifies the entire active site, because parts of it, you can inhibit the enzyme substrate binding, or it can destroy the ability of the active site to bind the substrate altogether. So you can make it partially bind, make it so it doesn't bind at all. It's still not going to work real well. All right, so heating is an effective mechanism for killing bacteria and viruses on surgical instruments. How does elevated temperature result in cellular death? Okay, we just talked about that, right? High heat denatures proteins, including the enzymes of living cells. If you kill the enzymes, the cells can't function. They can't continue to replicate. They can't do the things they're supposed to do. Okay, that's what keeps the cells alive, that keeps them going, is the enzymes allows them to create different metabolic factors that are keeping the organism alive. And heat denatures those proteins, so the living cells die on medical instruments, or viruses die also. Okay, so heating surgical instruments, an effective means of sterilization, if the heat treatment is carried out under conditions and temperature and time that are sufficient to kill microbial life forms. So you have to have a certain temperature and has to have a certain time. So as the pressure increases or temperature increases, it can decrease the time. And at 2 atmospheric pressure, which we build up our autoclaves to, okay, 20 minutes later, everything's dead. Okay. Regulation of enzyme activity. All right. One of the major ways that enzymes differ from non-biological catalysts is in the regulation of biological catalysts by cells. Okay, so it's going to regulate within the cell for catalyze that reaction. So some methods that we're going to use to regulate their enzymes, okay, are they produce the enzyme only when the substrate is present. This is common bacteria. So we have ways to modify how we're going to use these enzymes in the body. And it's not like in the lab where you want to make something, you pour a couple things into this, and you mix them together, and you heat up, and poof, it's going to go. The body can't do that. The body has to say, the body can't have the enzyme, like say, let's use pepsin. We can't have pepsin active all the time. If pepsin is active all the time, there's no food in the stomach, the pepsin will start eating the stomach, right? It's going to dissolve protein, which is bad for your stomach lining. So you only have to have that enzyme around when you need it, like all the enzymes. There has to be a regulatory system in your body, because if you turn everything loose and have no way of regulating it, it's going to destroy your entire body. Because that's what enzymes do. They destroy proteins, they destroy different things, and they reuse that stuff and put it in the body so we can use it again. So we have to have ways to modify, to regulate what we can produce and what we don't produce. Okay? In all things in the body works that way, just like insulin and glucagon. Okay, if there's sugar in the bloodstream, the body produces insulin. Insulin takes the sugar out of the blood. If the body's hypoglycemic, out of sugar, the body produces glycagon, which breaks down triglycerides and fats and puts sugar back in the blood. Okay? We have to have both ways to regulate things. We can't just turn something on and let it run wild. Okay? It doesn't work that way. It's not going to work that way. So one of the ways you can regulate is to produce the enzyme only when we need it. Okay, so if in an environment and all of a sudden there's food in the stomach, the body says, oh, there's food. And it triggers a nerve, it triggers a receptor and says, now there's food in the stomach, now make the pepsin. Okay? So you're only going to produce it when you need it. And the enzymes are short-lived. So when you don't need it, it's going to die off to stop being produced. Okay? Allosteric enzymes, which we'll talk about. Allosteric enzymes are ways we can modify, we can have an enzyme present all the time, but it's not active. We can always make it, but it's not active. Allosteric, we can activate the enzyme that's already there. Okay? And we have feedback inhibition. Okay, a feedback loop is basically a loop that says if something's being produced here, it's running around, and this is your product. When the product is produced here, the product comes down and shuts that down. So when you're producing the enzyme, now it's making the product from the enzyme substrate complex to the enzyme product complex, produces the product. Once the product's in the area, it shuts down the production of the enzyme. That's a feedback loop. It stops it from occurring once the product is being generated. So we can produce the enzyme when the substrate is present. We have allosteric enzymes, which we'll go over in just a second, how the different ones are activated. And we have a feedback inhibition, a feedback loop. Okay? And you can have what's called proenzymes. Proenzymes are zymogens. Zymogens are enzymes that are made and they're not active until they're modified, completely modified. Like cut into pieces to become active. Like that's how pepsin works actually. Okay? And we can have protein modifications. You can modify the proteins to make it work. So there's 5 different methods we can use to regulate that enzyme activity in the body. Okay, so enzyme activity is often regulated by the cell. The cells control the enzyme activity within it. Often the reason for this is to conserve energy. Okay, the cell's going to be using it. The cell's going to control what's there. If the cell runs out of chemical energy, it's going to die. Therefore, many mechanisms exist to conserve cellular energy. Okay, cell has some energy to function. There's a great waste of energy to produce an enzyme if there's no substrate available. So why would the cell sit there and make this enzyme if you're not going to use it? If nothing's there for it to work with? It's not. And if it's using all the energy to do that, it's wasting its energy and not be able to use it for cell division, mitosis. Okay, it doesn't have energy for that, then the cell's going to die off. Okay? So if the product of an enzyme catalyzed reaction is present in excess, so if you wanted to make, I'm trying to think of a good example. If you're taking your enzyme and your substrate and you're producing product and this product is just increasing, okay? The product is present in excess. It's a waste of energy of the enzyme to continue producing more than one product, right? So if the enzyme's working and making product and this product is building up, you want to stop this enzyme from working. Okay? Because there's already too much product there. Okay? You don't want to waste that energy. The simplest mechanism of enzyme regulation is to produce the enzyme only when the substrate is present. That's the easiest way to do it. The substrate is present, produce the enzyme. If it's not present, don't produce the enzyme. That's one way of regulating it. This mechanism is used by bacteria to regulate the enzymes needed to break down various sugars to yield ATP for cellular work. Okay? So the bacteria have no control over the environment or over what food sources, if any, might be available. The bacteria can't pull into an all-night diner and order whatever they want to eat, right? They don't have control over it. They're subjected to whatever's in the environment, that's what they're going to get. Okay, so whatever's going to be available to bacteria, that's what they have to use. It would be an enormous waste of energy to produce all of the enzymes that are needed to break down all the possible sugars, because you don't know which sugars are going to be available. Bacteria have no idea which sugars are going to be present in their environment. So you can't just make all the enzymes break down all the possible sugars. Okay? You only make the enzymes you need for the sugars you're exposed to at that time. Right? So the bacteria save energy producing the enzymes only when a specific sugar substrate is available. So when there's a specific sugar substrate in the environment, bacteria goes, oh, that's the sugar that's here. We need to break that down for energy. I need to produce this particular enzyme that's going to be the complementary structure to the sugar substrate present. Okay. Allosteric enzymes. Allosteric enzymes are effector molecules. They change the activity of the enzyme by binding at a second site. They do not bind to the active site, but they bind to a second site on the enzyme, and which then modifies the active site. Okay, so some effectors speed up enzyme action. Those are positive allosterisms. They make it go faster. Okay, some effectors slow down enzyme action. Those are negative allosterism. They make it go slower. So for allosteric enzymes, positive allosterism makes it go faster. Negative allosterism makes it go slower. Okay, so allosteric enzymes. These are effector molecules change the active site of an enzyme by binding at a second site. So they change the active site, so they change the activity of the enzyme by binding at a second site. They do not bind to the active site. Okay, they do not modify it directly, but they bind to another site. So if this is the enzyme, okay, a positive allosterism we talked about, the effector binding converts the active site to an active configuration. Negative allosterism, effector binding converts the active site to an inactive configuration. So positive allosterism, it goes faster. Negative allosterism, it goes slower. All right, so this is your enzyme right here. This is your enzyme. This is the effector binding site. This is the active site where the substrate binds to. Okay, there's your active site. Now, if so the substrate binds to the enzyme, you get your enzyme substrate complex, which you have here. Then you get your, this turns it to an enzyme product complex, and then the product is released. The substrate turns into the product. Okay. Now, when you release the product is in the solution. It comes back, and this product binds to this effector site right here. When the product binds to the effector site, it changes the shape of the active site, and the active site is now closed. Okay, so you have your enzyme, you have your substrate. As you produce product, the product goes back and binds to a second site. When it binds to that second site, it modifies the shape of the active site and basically closes it down. That's allosteric enzyme. When there's a second site where something binds to. And it's like a feedback loop. The more products produced, the more it binds to the effector site, shutting down the active site, making the enzyme inactive. Okay. So here's your substrate, and this is your active site of your enzyme. Okay. This is an allosteric activator. So when this binds to that allosteric site, the active site is open. If the active site is open, the substrate molecule can go whoosh, and come on in and become modified. Okay. Now, if this is an allosteric activator, an allosteric inhibitor, if that binds to this secondary site, it basically changes the active site. So now the substrate cannot fit in there. Okay. That's allosteric inhibitor. It shuts down the enzyme from working. Okay. And a lot of times, this allosteric inhibitor is the product itself. So when the product's being produced, it goes out and shuts the enzyme down because the product's already being produced, has enough in there. Okay. But this is allosteric enzyme activity. Allosteric is when it binds to a second site, and that binding modifies the active site. Okay. If it's a positive, it modifies the active site, so the site will accept the substrate. If it's a negative allosterism, it's negative allosterism, it binds to the secondary site and causes the active site to not be able to accept the substrate, to change its shape so the substrate cannot fit. That's allosterism. Okay, allosteric metabolism. So the third reaction of glycolysis, which we're going to study in the next chapter. Okay, glycolysis places a second phosphate group on fructose 6-phosphate. The phosphate groups are used for energy transfer in forming ATP, adding diphosphate, adding triphosphate, and that's a storage form for energy in the body. So phosphate groups are important in how they transfer them. And we're going to have to keep track of those as we learn glycolysis. Okay, so ATP is a negative effector. Okay, think about that. ATP is a negative effector because if you have ATP present, you don't need it anymore. It's already here. We have the energy that we need. So if there's ATP in excess in the cell, it's going to be a negative effector. It's going to stop the production of glycolysis because you have enough ATP. AMP is a positive effector. ATP is adding triphosphate, which is 3 phosphate groups. AMP is adding monophosphate, which is 1 phosphate group. So AMP goes to ADP and goes to ATP. So if you have extra AMP floating around, you need to make ATP. So it's going to be a positive effector. It's going to make more ATP. If you have enough ATP floating already, it's a negative effector. It's going to shut down the production so you don't waste more energy trying to produce something you already have. Okay, so ATP is a negative effector, AMP is a positive effector of the enzyme phosphofructokinase. Phosphofructokinase adds a phosphate group to fructose 6-phosphate. It allows you to transfer to your adenine molecules. Okay, so here is your fructose 6-phosphate and then fructose 1,6 biphosphate. So you can see this fructose group has 1 phosphate group plus ATP takes energy and it adds a phosphate group to fructose 1,6 biphosphate. And the enzyme that does it is phosphofructokinase. That's the enzyme that allows that to happen. Okay. So it allows you to store and move phosphate groups. Okay, so you go from fructose 6-phosphate to fructose 1,6 biphosphate. ATP causes that reaction to slow down if it's already present. AMP is a positive, wants more of it to occur. So you get more reaction with transitive phosphate groups to make more ATP. Right? So phosphofructokinase is sensitive to both positive and negative allosterism, which we just talked about. Okay? So when ATP is present in abundance, a signal that the body has sufficient energy, it binds to an effector binding on phosphofructokinase, and it's going to shut it down. Okay? This inhibits the activity of the enzyme and slows down the entire pathway because you already have enough energy. You don't have to transfer the phosphate groups to keep that energy going. Okay, an abundance of AMP, okay, which is a precursor to ATP, is evidence the body needs to make more ATP because ATP breaks down to ADP and AMP. Okay, so if you have AMP in abundance, then you're telling the body, hey, you need more energy. So AMP is also going to be an allosteric, binding allosterically. Okay, so AMP binds to effector site on the phosphofructokinase, and the activity is increased. Okay, it's a positive allosteric effect, speeding up the reaction and then the entire pathway. So more phosphate groups are transferred. Okay, more energy is created. Feedback inhibition. So now we go back and it's going to be like a feedback loop. Okay, allosteric enzymes are the basis for feedback inhibition, like we talked about. Okay, with feedback inhibition, a product late in a series of enzyme catalytic reactions serves as an inhibitor of the previous allosteric enzyme early in the series. So if the enzyme is catalyzing reaction, the products produced at the end are going to come back and stop the series, the enzyme from being active. So once the product is formed, all that means is you're in feedback inhibition. Once your product is formed, it's going to come back and stop the reaction from producing more products because we have enough of it. That's feedback inhibition. That's your feedback loop. So if you have the enzyme, A goes to B, goes to C, goes to D, goes to E, goes to F. A is your beginning. You have enzyme 1, enzyme 2, enzyme 3, enzyme 4, enzyme 5. F is your product. F is going to come back once it's produced and stop enzyme 1. Okay? It's going to inhibit enzyme 1 from, it's going to alter the active site and stop the active site from accepting the substrate. So it basically starts making more product that you don't need it. Okay. So in this example, product F serves to inhibit the activity of enzyme 1. That's what a feedback negative feedback loop or feedback inhibition is. Okay, so product F acts as a negative allosteric effector. So F here is a negative allosteric effector. It's going to bind to a secondary site on enzyme 1 and cause an alteration of the active site on enzyme 1. So the substrate A cannot bind to the active site and be produced into B and so on. Okay. Now, it affects enzyme 1. Why couldn't it affect enzyme 5 and do the same thing? Because then, I mean, it would still effectively stop you from making the enzyme, but then you would still make enzyme at B and C and D and E using all that energy. So it's going to make, it makes more sense for it to stop it, where you're going to conserve the cell's energy, which it needs to survive, okay, at the first possible step. And that's what it does. It goes back to the first enzyme. Okay. So in the concentration of F, okay, when the concentration of F goes down, it will dissociate from the effector binding site. When that occurs, the enzyme is once again active. So the feedback inhibition is an effective metabolic on-off switch. So when F came back, okay, so F was coming back here. Now, if you use up all the F and it runs out, then the F does not bind to the E1 here. It opens up the active binding site and the reaction occurs again to make more F. So basically, if F is present, it turns the switch off. If F is not present, the switch gets turned on. So it's basically just an on-off switch that occurs. So proenzymes. A proenzyme is an enzyme made in an active form. A proenzyme, an enzyme made, is an inactive form. Okay. So a proenzyme is the enzyme is made, but it's inactive. The active site is non-functional, but the enzyme is made. Okay, that's a proenzyme. It's converted to its active form when it needs to work. It's converted by proteolysis, hydrolysis of the enzyme, removing water from it, bonding something to it, and then it becomes active. Okay. Or it can be activated when needed active site in the cell. They can activate directly at the active site. Okay. Pepsinogen, we talked about. Remember pepsin in the stomach? Pepsinogen is synthesized and transported to the stomach. Once it's in the stomach, it's converted to pepsin. Now, think about it. Pepsin dissolves proteins. That's its main function. Do you want that floating through your body, getting to the site where it needs to work? No, you don't. Okay? You don't want to destroy everything in your body. Okay? You want pepsinogen is produced, it's inactive. So now it transmits through the body and it ends up in the small into the stomach. Once it's in the stomach, then it's converted to pepsin where it becomes active. Okay? Because you don't want that active everywhere else. So that is what's called a proenzyme. A proenzyme is made in the inactive form. Then you allow it to go to where you need it to, or something triggers it. When you want to become active, then it becomes active. Okay, this is some of the proenzyme digestive tract. Proteolastase converts to elastase. Okay, and it's activated by trypsin, the amino acid. Trypsinogen, okay, is activated to trypsin. Chymotrypsinogen A is activated to chymotrypsin. Pepsinogen, we just talked about. The activator happens to be the acid pH. Remember, stomach has a pH of 2, and the presence of pepsin. And then pepsin, because pepsin's present in the stomach, then you have the enzyme pepsin being active. Procarboxypeptidases. Trypsin, again, is the activator because trypsin found in the stomach. Carboxypeptidase A and carboxypeptidase B become from the procarboxypeptidases. Okay? So what you need to know is a proenzyme is made, it is inactive. It becomes active at where the active site is by a certain factor. With pepsinogen, goes to pepsin in the presence of acid pH and the presence of pepsin originally being there. Protein modifications. We can modify enzymes, and protein modification of a chemical group is covalently added or removed from the protein. So we're modifying the protein. Okay, covalent modification either activates or turns off the enzyme. Okay? So you either take it, activate it, or turn it off when you're going to modify it. The most common form of protein modification is addition or removal of a phosphate group. Okay, that's the most common form of protein modifications. Phosphate groups are used to transport making energy, ADP to ATP, etc. Phosphate groups are energy stores that we use to transfer when we break those bonds. We produce energy. Okay? So the most common form of modifying a protein, okay, is the adding or removal of a phosphate group. This group is located at the R group with a free OH, okay, of like the amino acid serine, threonine, tyrosine. Okay, covalent modification of enzyme structure is catalyzed by other enzymes. So other enzymes catalyze the structure of that enzyme. Protein kinases add phosphor groups to a target enzyme, while phosphatases remove phosphor groups to target enzymes. So kinases add phosphor groups. Okay, proteases remove phosphor groups from enzymes. Now, these we're talking now around covalent bonds. We're not talking the tertiary structures anymore. We're talking covalent bonds, strong bonds that are binding them together. So that's why we need enzymes to modify them. For some enzymes, it's phosphorylated. For some enzymes, when it goes to the phosphorylated form, and when phosphate's added to it, then it becomes active. Okay. For instance, in adipose tissue, in fat tissue is triglycerides. That's how your body stores fat, is in your triglycerides. Okay. That is our energy storage. Fat is our energy storage for our body. Okay. When you eat too much food and we can't use it in energy right away, it's stored as fat in your body. It's stored for energy for later on. It's stored in the form of triglycerides. Okay, so the adipose tissue is phosphorylation activates the enzyme triacylglycerol lipase that breaks triglycerides down to fatty acids and glycerol. When it breaks down to fatty acids and glycerol, energy is released. Okay, you have phosphate groups that can be added to this. Okay, so phosphorylation activates. So when you're phosphorylating it, it activates the triacylglycerol lipase, which breaks down the triglycerides to release fatty acids that can be used and glycerol to make energy, to make ATP. Okay. Glycogen phosphorylase is an enzyme involved in the breakdown of glycogen. It's also activated by the addition of phosphoryl groups. So you want to break down glycogen. Okay. It's activated when you get phosphoryl groups activated to it, and you activate glycogen phosphorylase, and that starts the breakdown of glycogen. Okay. Some enzymes, phosphorylation inactivates the enzyme. So certain enzymes, when you add phosphate groups to it, it activates it. Other enzymes, it inactivates it. This is true for glycogen synthase, enzyme involved in the synthesis of glycogen when you want to make glycogen for storage. Okay. So when the enzyme is phosphorylated, it becomes inactive. The convenient aspect of this type of regulation is it's reversible. Okay, phosphorylation is reversible. You can phosphorylate or you can dephosphorylate depending on the enzyme. And a second on-off switch again, just like you did before. Turn the enzyme on or turn the enzyme off. An enzyme can quickly be turned on or off in response to environmental or physiological conditions by the level of phosphate in the environment. All right, inhibition of enzyme activity. How can you stop enzyme activity? Remember, you turn it on, you gotta be able to turn it off. Chemicals can bind to enzymes and eliminate or drastically reduce catalytic activity. Catalytic activity is your ability of the enzyme to break down your substrate into a product. Okay. You classify enzyme inhibitors on the basis of reversibility and competition. So how it inhibits the enzyme from working can be because it could be reversible, or it can compete with something at that active site. It's binding to the same site now. So irreversible inhibitors. Okay. Irreversible inhibitors bind tightly to the enzyme and prevent formation of the enzyme substrate complex. Okay, they bind and they shut it down completely. It's irreversible. Reversible competitive inhibitors. Structure resemble the substrate and bind at the normal active site. So these inhibitors, they're not binding at a secondary site. They're binding at the active site. So irreversible inhibitors bind so tightly they can't be kicked off. Reversible competitive inhibitors are now in competition, which means they're going to bind to the active site. And if they're on the active site, then the regular structure can't get at it. Okay? But it comes off and on. So there is some competition now between which one's going to be used at active site. Okay? It's kind of like how carbon monoxide poisoning works. Okay? One form of suicide: lock yourself in the garage, turn your car on, let the engine run, and produce carbon monoxide. Okay, carbon monoxide competes with oxygen at hemoglobin for the red blood cell. Okay, the red blood cell binds oxygen, carries it to the body, provides oxygen for your body. Carbon monoxide binds to the same site oxygen does. Okay, and it's a stronger bond. So when carbon monoxide bonds, it bonds in place of oxygen and doesn't let oxygen attach. That's why carbon monoxide poisoning is so deadly. That's what causes death, because it doesn't allow oxygen to bond to your red blood cell. Okay, so the treatment of carbon monoxide poisoning is hyperbaric oxygen chambers, or getting the person outside where there is no more carbon monoxide. That way carbon monoxide eventually breaks off and then more oxygen is replaced to it. But if carbon monoxide and oxygen, because you got to remember your garage is still filled with oxygen. Okay, there's still plenty of oxygen in there, but the carbon monoxide builds up to the point and it bonds more competitively than oxygen. And that's why you die from it. Okay, that's a competitive inhibitor. So reversible competitive inhibitors, they resemble the substrate and they bind at the normal active site. They do not bind at allosteric site. They bind at the active site and they compete for that active site. Who's going to bind there? Reversible non-competitive inhibitors usually bind to someplace other than the active site. Okay, and they come off and on. They don't compete. If they're competitive, they're binding at the exact same site, okay, as the substrate. So binding is weak, and thus inhibition is reversible. So you can have irreversible inhibitors that bind tightly. You can have reversible competitive inhibitors where they bond and they compete with the other inhibitors, or they can reversible non-competitive, they bond at a different site and shut the enzyme down that way. It's a weaker bond. So enzyme inhibitors are classified on the basis of whether the inhibition is reversible or irreversible, competitive or non-competitive. Irreversible is a strong bond that you're not going to break off. Irreversible means it can come off. Competitive means it's bonding to the same site. Non-competitive means it's bonding at a different site other than the active site. Okay. So reversibility deals with whether the inhibitor will eventually dissociate from the enzyme, releasing it in the active form. That's reversibility. So what's inhibiting it can eventually break off and causing the active site to become active again. Okay. Competition refers to whether the inhibitor is a structural analog or lookalike of the natural substance. So competition is both bind to the active site, and they both have similar shapes, like an analog. Okay, and it looks like the natural substance, so it binds to it. And therefore, there's competition for that binding site. If so, the inhibitor and the substrate will compete for the enzyme active site. So enzymes, neurotransmissions, and nerve agents. This is how things work in the body. Where do we have it? There we go. Transmission of the nerve impulses at the neuromuscular junction involve multiple steps. Okay, so you got to remember that you send a signal. Like say you want to pick up your pencil to take notes, or say you want to use your fingers to flip someone off that cuts you off in the freeway. Okay, it's the same thing. It's the transmission of nerve impulse to the neuromuscular junction. It means the nerve and the muscle innervate. Where the muscle innervates the nerve innervates the muscle. Okay. One of which is the activity of a critical enzyme called acetylcholinesterase. Acetylcholinesterase is a neurotransmitter. Okay, it catalyzes the hydrolysis of the chemical messenger, acetylcholine, and initiates the nerve impulse. So acetylcholinesterase is an enzyme that blocks or stops acetylcholine from triggering that nerve. It breaks down acetylcholine. Okay. The need for this enzyme activity becomes clear when we consider the events that begin when a message from the nerve cell and ends in the appropriate response by the muscle cell. So acetylcholine, the neurotransmitter, it's a chemical messenger. It transmits a message from the nerve cell to the muscle. So this is your nerve coming down, and it stops right here. And this is your muscle fibers. And this is the space between them. Okay. So this sends information across that gap to the muscle cell and makes your muscle cell contract. So basically the nerve comes down, acetylcholine is released, and the muscle contracts. Now think about it. If acetylcholine stays there, acetylcholine binds to receptors that causes the muscle to contract. Why would the muscle stop contracting? If acetylcholine is there, the muscle is going to contract. So you need something to get rid of the acetylcholine once the muscle contracts, because you don't want your muscles to contract and stay contracted. That's tetanus. That's dead. That's instant death. And if your heart contracts and can't relax, it's not going to beat again. Okay. With tetanus, sometimes people can spasm, and they can spasm and contract so bad they'll actually snap their spinal cord. They'll actually break their own spinal cord from their muscles. Okay? So you have to have something that stops acetylcholine once it's done its job. And that's where acetylcholinesterase comes in. Acetylcholine is stored in the membrane-bound bags called synaptic vesicles in the nerve ending. Okay? And it's probably a better picture than I can draw. But this is the nerve coming down here. Okay? And it releases acetylcholine from these little vesicles into this gap. This gap here is a neuromuscular junction. These receptors bind to the acetylcholine. Acetylcholine causes calcium to be released, and it causes muscle here to contract, to shorten. Again, muscles can only move one direction. All they do is shorten. They can't do anything else. So the muscle shortens. Okay. Now, if that acetylcholine keeps in that junction, that muscle is going to contract, 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 and never release. Okay. So that's we have acetylcholinesterase. That's the enzyme. Acetylcholinesterase breaks down acetylcholine and allows it to be reused and be picked up and made back into acetylcholine again. But it breaks it down so it does not cause continuous contraction of the muscles. So the nerve impulse carries acetylcholine to be released from the synaptic vesicles. Acetylcholine diffuses across the synaptic cleft, across that gap, and binds to specific receptors on the postsynaptic membrane, on the muscle, on the membrane of the muscle cells. A channel opens up, allows sodium ions to flow into the cell, potassium ions to flow out of the cell. Okay. This results in muscle contraction. Okay. Any acetylcholine remaining in the synaptic cleft is destroyed by acetylcholinesterase. Acetylcholinesterase, that's the enzyme. Remember, ASEs are enzymes. Okay. Acetylcholinesterase breaks down acetylcholine. It terminates the stimulation of the muscle cell, which is just as important as the initial contraction. Once you contract it, you have to be able to stop it and let that muscle relax again. Okay. So acetylcholinesterase comes into play. Okay. The arrival of the nerve impulse at the end plate of the nerve axon causes an influx of calcium. You'll learn in anatomy that there's two things required to contract a muscle: calcium and ATP. Those two things you have to contract a muscle. Okay, so calcium comes across from the nerve. This causes acetylcholine-containing vesicles to migrate to the nerve cell membrane that is in contact with the muscle cell. Okay, this is your presynaptic membrane. This is the membrane of the nerve cell that is on the first side of the junction before it gets to the muscle. All right. Vesicle fuses with the presynaptic membrane and releases a neurotransmitter into the space. So acetylcholine is released into the space between the nerve and the muscle. The acetylcholine diffuses across the nerve synapse, okay, and binds to acetylcholine receptor protein in the postsynaptic membrane of the muscle cell. Okay. This receptor then opens up pores in the membrane through which sodium and potassium ions flow into and out of the cell. Okay. Generates a nerve impulse and causes the muscle to contract. So if acetylcholine remains in the nervous junction, it will continue to stimulate muscle contraction. So it can't stay there because that is not a good thing. You have to be able to stop that contraction once you start it. Okay, to stop this continuous stimulation, acetylcholine is hydrolyzed, okay, destroyed by acetylcholinesterase. There's your enzyme. That's the importance of the enzyme. Acetylcholinesterase breaks down acetylcholine. Okay, when this happens, the nerve impulse stops and the muscle can relax. Inhibitors of acetylcholinesterase are used as poisons and as drugs. If you want to poison somebody, okay, you usually want to initiate their demise, right? That's what you do when you poison somebody. If you put a drug in there that binds to or stops acetylcholinesterase from working, what's going to happen? All the muscles in the body are going to contract and there's no way they're going to ever relax. They're going to what's called tetanus. Okay. So among the most important inhibitors of acetylcholinesterase are a class of compounds known as organophosphates. Organophosphates are deadly poisons. They work really well. One of these is sarin gas. Sarin gas was released in, where was that? Was it Japan or London? It was either Japan or London. They released sarin gas into the subway system. Gosh, I forget what it was. Okay, sarin gas, it's airborne. It's an acetylcholinesterase inhibitor. So it stops acetylcholinesterase from working. So the muscles of the body contract and they can't uncontract. So the heart contracts to beat and it can't relax. Can't feel the blood anymore. Okay, all your muscles contract. All your muscles contract in your body and they can't release. That's sarin gas. It's an organophosphate.
It was in Tokyo, by the way.
Was it Tokyo?
Mm-hmm.
Okay.
Yeah.
I knew it was Tokyo, Japan or London. Okay. That was in Tokyo. I was in Tokyo in the subway. It was a sarin gas attack. And the name of it is isopropyl methylfluorophosphate. Okay. You know what isopropyl is? You know what methyl is? You know what fluoro is? And it's a phosphate attached to it. So it's a very simple poison. Okay. Sarin forms a covalently bond intermediate with the active site of acetylcholinesterase. So now think what it's saying. Acetylcholinesterase, which we need to stop the muscle contracting, it forms a covalent bond at the active site. Covalent bond is a strong, irreversible bond. It is not the bonds that we use for bonding our substrate to our substrate to our enzymes. Those are our weak bonds: hydrogen bonds, diphosphate, disulfide bonds, salt bridge bonds, hydrophobic interactions. Okay, this is covalently bonded to the active site of acetylcholinesterase, which means acetylcholinesterase is not going to work. Period. So when you release acetylcholine to contract your muscles, it's going to contract and never be able to be released. Okay, it is an irreversible competitive inhibitor. It's irreversible because it is a covalent bond. It's competitive because it bonds to the active site. Okay, that's what those terms define. So irreversible means it is a covalent bond, not a regular weak bond like it can be reversible. And competitive means it is bonding to the active site of the enzyme. Okay, and that's what it looks like. Here's your isopropyl group, there's your phosphate group, your fluoride group, and your methyl group. Okay, that's sarin gas. So sarin nerve gas, okay, basically what it says, we'll sum it up right here. This is your junctions here. This is your impulse, your nerve coming down. Okay, this is your electrical signal. Fused with these cells, and this is your acetylcholine. Acetylcholine comes in here, it fills this gap, okay, and it binds to these receptors. These receptors cause calcium to be released into the muscle, and this muscle now contracts, shortens. Oops, oh, I went backwards. Where did I go backwards here? There we are. Came back. All right. So once acetylcholine's in the gap, it's going to cause a contraction. Now, the acetylcholinesterase binds to that. Okay, here's your acetylcholinesterase, binds to that and breaks it down into acetate and choline, okay, and stops the contraction. Sarin gas comes along, and sarin gas binds to the acetylcholinesterase, and now it binds to the active site. So the acetylcholinesterase cannot bind to the acetylcholine and break it down into acetate and choline. Okay. So when sarin gas is present, the muscle contracts and cannot release. Okay. End result with sarin gas is overstimulation of muscle, causing shutdown of the respiratory system and all your other bodily functions. It's not a pleasant way to die. Every system in your body shuts down. So the covalent intermediate is stable, and acetylcholinesterase is therefore inactive, no longer able to break down acetylcholine into acetyl and choline. So nerve transmission continues, resulting in muscle spasms. Every body in your muscle spasms. Okay, you ever have a charty horse cramp, your leg cramp at night? You know how bad that hurts? Imagine every single muscle in your body doing that. It's not a pleasant way to die. That's what sarin gas causes. Death may occur as a result of a laryngeal spasm, where your larynx, which is your tube between your oral cavity and your lungs, shuts off. You can't get any oxygen in. Those muscles can't relax. Okay, antidotes for poisoning by organophosphates, which may include many insecticides and nerve gases, have been developed. Okay, if someone can poison you, someone can unpoison you, basically. Okay, these organophosphates are present in a lot of insecticides that we spray and a lot of nerve gases. Okay, the antidotes work by reversing the effects of the inhibitor. Okay, one of these antidotes is known as PAM. It's for pyridine, aldoxamine, methionine. PAM is an antidote to sarin gas, to the organophosphates. This molecule displaces the organophosphate group from the active site of the enzyme. Okay, so that's the sarin gas binds, covalently binds to the acetylcholine esterase. So what PAM does, it binds to that and it unleashes, okay, the sarin gas molecule from the acetylcholinesterase, reopening up the active site. Okay, and that's what it looks like. That's what PAM looks like. So it binds to another site that allows that to be reversible bond now and breaks that covalent bond that's been established. Oh, Tokyo, here it's right. Let's see in my notes. I should have read ahead, huh? Okay, on Monday, 20 March 1995, 5 members of Amu Shinrikyo launched a chemical attack on the Tokyo metro system, one of the world's busiest commuter transport system at the peak of the morning rush hour. Okay, the chemical agent used was liquid sarin gas, contained in plastic bags, which each team was wrapped in a newspaper. Okay, each perpetrator carried 2 packets of sarin, totaling approximately 900 milliliters of sarin, except Yashi Hasuko, who carried 3 bags. He must have been the leader. So a lot of the sarin gas is very deadly. A single drop of sarin gas the size of a pinhead can kill an adult. So 900 milliliters is a tremendous amount of sarin gas. They can kill a lot of people. It's very, very potent. In 5 coordinated attacks, at least dozens of people were killed, 50 were severely injured, and thousands had temporary vision problems. As the sarin gas accumulated in the eyes. Carrying the packets of sarin with umbrellas with sharpened tips, the perpetrators boarded their appointed trains. At prearranged stations, the sarin packets were dropped and punctured several times with the sharpened tip of the umbrella and released the gas into the air. The men got off the train and exited the station to meet his accomplice with a car. Okay. By leaving the punctured packets on the floor, the sarin was allowed to leak out and evaporate into the train car and stations. Okay, as it evaporated, formed the gas. That's where the people put it there weren't killed. Okay. The sarin vapor was inhaled, causing the reaction. The principal chemicals of curare Same way. That's how sarin gas works. Now, curare, okay, are alkaloids that affect your neuromuscular junction also. They affect it in a different way. These drugs are employed as a relaxant of skeletal muscles during surgery to control convulsions. Okay. Research has shown that curare causes a weakening and paralysis of skeletal muscles by interfering with the transmission of nervous impulses between the nerve axon and the contraction mechanism of the muscle cells. So now curare gets in there and stops the contraction, the exact opposite of sarin gas that works on the enzyme. Okay, the alkaloid interferes with the activity of acetylcholine to depolarize the cell's end plate at the surface where it's functioned, thereby blocking the neurotransmuscular junction. So curare blocks acetylcholine from getting to the receptor. Okay, it depolarises the cell at its end plate. So in other words, it can't bind to the receptor; it's not released. So the curare takes its place. Now we use it in surgery, and it causes paralysis. Okay, it stops the acetylcholine from transmitting across the junction into and stimulating the muscle to contract. So the patient holds still for surgery, which is a good thing, right? What's the problem with this picture? Curare paralyzes you, does nothing for pain control. So you are completely paralyzed. You cannot move anything, and you can feel everything. That's the problem. Okay, you paralyze all the muscles, but you have not paralyzed your nervous system. You paralyze the release of acetylcholine. You have not stopped your nerves from firing. So if you put someone on curare, they will feel every cut you make, every single one. Okay. Remember that. You go into medical school, you have to make sure you understand exactly what's being inferred chemically inside the body. Okay. So it will paralyze you by stopping acetylcholine, but every nerve in your body will still feel. You just won't be able to move anything. Okay, death from curare is caused by asphyxiation because the skeletal muscles become relaxed and they can't move. So you die of asphyxiation. Okay, the poison only works in the blood. Poisoned animals have no harmful effects on human. Okay, curare is used in Africa, South America, as a poison in darts, using to kill animals for food. Okay, curare is in their blood. It will not hurt you if you ingest it orally. Okay, it has to be injected into the blood to work. Poison darts, things like that. Okay, its vapors are not poisonous, although many natives believe that they were poisonous, so they were careful when they made it. But the vapors of curare are not poisonous. It has to be injected into the bloodstream to stop the acetylcholine. In 1811, Sir Benjamin Brodie noted that during curare poisoning, the heart continues to beat even after breathing stops, which means the heart function is not stopped by curare. That's why we were able to use it in surgery, okay? Because it does not stop the heart from beating. Okay. The horror of curare poisoning is the victim is very much awake. That's like we said, aware of what is happening until the loss of consciousness from lack of oxygen. Okay, so if you do curare poisoning, your patient can feel every single thing that you do, and they cannot move. They can't even blink an eye. Again, not a good way to die. Consequently, the victim can feel the progressive paralysis, but can do nothing to call out or make anybody aware of it. If artificial respiration is performed throughout the ordeal, the victim is going to recover with no ill effects. So if someone has curare, you keep them on a respirator, they'll do just fine once the curare wears off. Curare is still used in many chronic situations like muscle trauma or spasms, acute arthritis, poliomyelitis, as well as in surgery. But you have to use other drugs in combination with it. You have to use analgesics. You have to use something that's going to kill the pain, or the patient will not like you, or they'll probably die on the table too from the pain. Sir Walter Raleigh and several others who explored South America reported on curare and its effect. The usual method to prepare it was to combine young bark scrapings of strychnose, which is a mini sperms with other clean plant fragments, and sometimes snake venom or venomous ants. Okay. The mixture is then boiled in water for about 2 days, then stained and evaporated to become dark, heavy, viscid paste with a very bitter taste. Now, they tell you can't get it by eating it, but I'm not going to taste it. I don't know about you, but I'm not going to be the one that says, oh, that tastes very bitter, so I can report on that. Yeah, let someone else do that part. People make mistakes. The potency would be tested by counting the number of leaps a frog would take after being pricked. So they prick it with curare, see how many jumps it would take, and that's how potent it was. 1 jump, 2 jump, 3 jumps. 1 jump was really potent. 3 jumps was not as potent. That's how they determine the strength of the curare mixture. Okay. Darts were tipped with curare, accurately fired through blowguns made of bamboo. Death for birds would take 1 to 2 minutes. Small mammals, up to 10 minutes. Large mammals, up to 20 minutes with curare, as they were poisoned. Okay. Curare was too expensive and scarce to be used in warfare. So they never used it battling between tribes. They never used it against the other humans because it was too expensive to waste on people. They had to use it for the food supply. Interesting. All right, irreversible inhibitors. So we talked about reversible and irreversible, competitive and non-competitive. Irreversible means it's forming what kind of bond? It's forming a covalent bond. It's forming a strong bond. Okay, so irreversible can be competitive or non-competitive. Competitive, it binds at the active site. Non-competitive is binding at a site other than the active site. So irreversible inhibitors, okay, they bind tightly to the enzyme and may form a covalent bond. Okay, binding the inhibitor to one of the R groups of amino acid in the active site. Okay. Remember, irreversible means it's binding extremely tightly. It's going to form a covalent bond. It's not going to come off. It's not forming one of our weaker bonds. It can bind to the active site. This binding may block the active site so that the enzyme substrate complex cannot form. Remember, if you have the active site, so this is your enzyme right here, and this is your substrate that's going to fit inside of it here. Okay, if this bonds right here, okay, this ain't going to fit anymore. It's not going to make it. Okay, so it blocks the active site, so the enzyme substrate complex cannot be formed now. Alternately, an inhibitor may interfere with the catalytic group of the active site, eliminating catalysis. So it might eliminate the whole site altogether, and you'll have no catalytic reaction whatsoever. Okay, but the group that's causing it to the catalysis may interfere there, and then you can have no enzyme function whatsoever on it. Irreversible inhibitors include arsenic. Arsenic is a rat poison. Okay. It's an irreversible inhibitor. Snake venom. That's why it's so deadly. Nerve gas, the organophosphates we just talked about, sarin gas. Okay. Those are all irreversible inhibitors. Reversible, competitive inhibitors. Reversible means it's bonded by a weak bond, not covalently. Competitive means it's binding to the active site where the substrate has to form. It's competing with it. Okay, so reversible competitive enzyme inhibitors are also called structural analogs. Structural analogs because they have the same structure as the substrate. An analog is something that has the same structure as your original, but it's just a copy of it. So it's called a structural analog. Okay. Molecules that resemble the structure of the substrate and charge distribution of a natural substance for an enzyme. So whatever's going to bind in the enzyme, it mimics the shape and the charge distribution, and it binds to the active site in place of the enzyme. So resemblance permits the inhibitor to occupy the enzyme's active site. If it occupies the enzyme's active site, the normal substrate can't get to it. Okay, once the inhibitor is at the active site, no reaction can occur. Okay, the enzyme activity is inhibited because it blocks the active site. So the substrate can't fit in there. So no reaction is going to occur. Okay. Inhibition is competitive because the inhibitor and the substrate compete for the binding site, for binding to the active site of that enzyme. That's what's called competitive inhibition. It's reversible because it's not forming a covalent bond. It's forming a weaker bond, so it can be convinced to come off. The degree of inhibition, okay, how much is inhibited, depends on the relative concentrations of enzyme and inhibitor. Okay? If there's a lot of inhibitor around and little enzyme, it's going to block it completely. If there's a lot of enzyme around and a little bit of inhibitor, there's be plenty of enzymes for the other ones to work. Okay, so it all depends on how much of the concentrations are in the solution. Okay, reversible competitive inhibitors. This is your normal substrate. Okay, this is a competitive inhibitor, has a similar shape, similar charge. So they're going to go after the same enzyme site. So both molecules compete for this site right here. This is the active site of the enzyme. Okay, if the normal substrate gets here first, the enzyme substrate complex is formed and the reaction proceeds and produces a product. If the competitive inhibitor gets there first, this is formed. Okay, this blocks the enzyme from getting there. Okay, the competitive inhibitor is bound to the active site and it blocks the enzyme. So no reaction occurs. Okay, that's reversible competitive inhibition. Okay. Sulfa drugs work like that. Sulfa drugs are the first antimicrobials to be discovered. They are competitive inhibitors of bacterial enzyme needed for the synthesis of the vitamin folic acid. That's what sulfur drugs do. They're a competitive inhibitor, okay, for a bacterial enzyme. So it works on bacterial enzyme that makes folic acid. Folic acid is a vitamin required for the transfer of methyl groups in the biosynthesis of methionine and nitrogenous bases required to make DNA and RNA. So if we're blocking folic acid, the bacteria is not going to be able to make DNA and RNA, which means it can't replicate, which is a good thing, right? Humans cannot synthesize folic acid and must obtain it from our diet. So if we take sulfa drugs, we don't have an enzyme that makes folic acid because we can't make it anyways. So it's not going to affect us. It's only going to affect the bacteria because they have the enzyme that makes folic acid, whereas we don't. Okay? We have to get folic acid in our diet. We can't make it in our bodies. So bacteria must make folic acid because they cannot take it in from the environment. So if we give them something, sulfa drugs, it blocks the enzyme, okay, competitively inhibits the enzyme for folic acid. Okay, they cannot make folic acid. If that enzyme is stopped, they can't make DNA and RNA, and they can't replicate, they die off. Para-aminobenzoic acid is a subject of an early step in folic acid synthesis. Let me get rid of that thing so it doesn't block your screen. There you go. Okay, that's para-amino. Remember para? Ortho. Remember the jokes? Ortho, meta, para. Okay, so it's para-amino. There's an amine group and the para position of benzoic acid, which I'll be able to should do that round, right? So there's your amino. Now it's going to cover it. There's your amine group right there. This is your benzoic acid. So para is just where it's located on the ortho, meta, para position. So para-aminobenzoic acid. Okay, that's what you use to first make folic acid. Okay, sulfa drugs are structural analogs of para-aminobenzoic acid. The competitive inhibitors of the enzyme that uses para-aminobenzoic acid as its normal substrate. So it inhibits, it's a competitive inhibitor for the enzyme that uses the para-aminobenzoic acid as the substrate in the production of folic acid for the bacteria. Okay. So pyrimidine acid on the left and sulfonamide, the sulfur enzyme on the right. You can see it has similar shapes. It can bind into something that has a similar area to it. So it would be a competitive inhibitor binding to the active site on the enzyme that catalyzes the reaction for folic acid in bacteria. If the correct substrate, pyrimidine benzoic acid, is bound by the enzyme, the reaction occurs and the bacteria lives. Okay. If the sulfa drug is present in excess over the pyrimidine benzoic acid, it binds more frequently to the active site of the enzyme. Okay. No folic acid will be produced. Bacteria cell cannot make DNA and RNA, and the bacteria cell will die. That's our first antibacterial medications work. That's your sulfa drugs. Now, the only reason sulfa drugs aren't used much anymore is there's a lot of people who are allergic to sulfa drugs, high amount. So sulfa drugs is not usually one of our first choices anymore because of the high incidence of allergic reactions in your patients. Okay. Because we obtain our folic acid from our diets, sulfa drugs do not harm us. Okay, because we're not relying on pyrimidine benzoic acid to form the folic acid. We have to get it from our diets anyways. Bacteria, so we selectively kill bacteria. That's why we want antibiotics to work. We want to only kill bacteria and not human cells. You want to use what's called the magic bullet, a silver bullet. You want to get it there, only kill the bacteria cells and not affect human cells. And that's how that works. Although bacteria infection was a major cause of death before the discovery of sulfa drugs and other antibiotics, death caused by bacterial infection is relatively rare at present. And yes and no, we're starting to get some super infections now, MRSA, things like that. We're starting to get some really nasty bacterial infections that we cannot fight. I mean, we give them antibiotics to our cattle, we give it out every time someone has a runny nose, a virus. Antibiotics do nothing against the virus. And what we're doing is we're isolating bacteria that are resistant to the antibiotics that we have. We give them to the cattle. What's happening is they're selecting antibiotics that are going to live in the presence of these antibiotics. Okay? They're mutating to fight against it. So a lot of antibiotics aren't working anymore. And we're getting these super infections which can't be treated. And now we got reversible non-competitive inhibitors. So reversible means we're not forming covalent bonds. We're using our weaker bonds to form. And we're not binding to the active site. The competitor is not, it's not binding the active site where it's causing the effect, like an allosteric enzyme. So reversible non-competitive enzyme inhibitors bind to R groups of amino acids or to the metal ion cofactor. They're not binding to the active site. This binding is weak. Okay, hydrogen bonds, salt bridges, hydrophobic interactions, things like that. Okay? It is not a covalent bond. Enzyme activity is restored when the inhibitor disassociates from the enzyme inhibitor complex. When the inhibitor breaks away, the enzyme function is restored. Okay, these inhibitors, okay, do not bind to the active site. That's the key. When you see the word non-competitive, the key is it does not bind to the active site. It's binding to an allosteric site. They modify the shape of the active site once they're bound somewhere else, just like an allosteric enzyme. Okay. I just read the chat too. What trees make toilet paper? Toilet trees. That's really good. I could use that in my next class. All right, so they don't bind the active site. They modify the shape of the active site. So once they're bound elsewhere in the structure, they modify so the active site cannot be accessed by the substrate. Just like a negative allosteric protein. Why are irreversible inhibitors considered to be poisons? They're irreversible. They bind to either the active site or they bind to a different site, an allosteric site, and they cause the enzyme to be non-functional by modifying the active site. If they're irreversible, it's poison. That's what a poison is, because you can't get it off. So therefore the enzyme is non-functional, just like sarin gas, just like curare. Irreversible inhibitors. So irreversible inhibitors bind very tightly, sometimes even covalently to an R group. If it's a competitive inhibitor, it will bind to an active site. If it's a non-competitive inhibitor, it will bind to not to the active site. Okay, they generally inhibit many different enzymes because they're not site-specific. The loss of enzyme activity impairs normal cell metabolism, resulting in death of the cell or death of the individual. Okay, explain the difference of an irreversible inhibitor and a reversible. Explain between an irreversible inhibitor and a reversible non-competitive inhibitor. Okay. An irreversible inhibitor does not come off, right? Okay. A reversible one does not make a strong bond, not like a covalent bond, and it's non-competitive, so it's not binding to the active site. So irreversible inhibitors bind very tightly, like covalently, to an R group in the active site, usually inhibit many different enzymes. Okay. The reversible non-competitive inhibitors form a weak bond and do not bind in the active site. They bind in an allosteric site, a site away from it. Okay. They modify the shape of the active site where they are bound to the other parts of the enzyme. Okay. And what is a structural analog? Structure means it has the same structure. An analog is the same shape and structure, but it's a copy. That's what an analog is. Okay. So a structural analog is something that will bind to the same active site because it has the same shape and charge. Okay. So a structural analog is a molecule that has a structure and charge distribution, okay, similar to that of a natural substrate. So it can bind to the active site in place of the substrate. That's a structural analog. It is very similar or has a similar portion of the molecule that is very close to the substrate, the original substrate. Okay. Generally able to bind to the enzyme's active site in place of the substrate. Okay. This inhibits enzyme activity because the normal substrate must compete with the structural analog to form the enzyme substrate complex. How can structural analogs serve as enzyme inhibitors? Just like we said, same thing, they're going to bind to the site. They're able to bind to the enzyme's active site. So they bind to the enzyme's active site because they look the same and they have the same charge distribution, so they can bind right to it. Okay. They inhibit the enzyme activity because your normal substrate has to compete with the structural analog, so it can try to bind to the site to form the enzyme substrate complex. If the structural analog is bound to the site, you cannot form the enzyme substrate complex because the analog's in the way. It's in the active site. Proteolytic enzymes. Yeah, 10 minutes, then we'll finish up. So proteolytic enzymes cleave the peptide bonds in proteins. Remember a peptide bond? You have proteins are made of amino acids linked together. The peptide bond is what holds those amino acids together. Proteolytic enzymes break those peptide bonds apart in your proteins. Okay, these enzymes break the peptide bonds and maintain the primary protein structure. Remember, the primary protein structure is just a list of enzymes that are linked, a list of amino acids that are linked together. So proteolytic enzymes break those peptide bonds. Okay, these enzymes specifically depend on a hydrophobic pocket. Okay, they have to have a hydrophobic pocket to get in there and break that bond. Okay, a cluster of hydrophobic amino acids brought together by the three-dimensional folding of the protein chain. Chymotrypsin, okay, cleaves the peptide bond at the carboxylic end. Remember, there's an N terminal and a C terminal. There's every amino acid has a nitrogen end and a carbon end. Okay, so it's all bonded by these NC bonds. Remember, there's N, C, C, N, C, C as it goes across. So this will cleave it. Chymotrypsin cleaves the peptide at the carboxylic end. So the carboxylic end, that's where it's going to cleave it. Remember, all amino acids have that same structure. Remember, this was your hydrogen, and then this is your R group. This is your hydrogen, this is your R group, this is your carbonyl, and this is your nitrogen. Okay. So it's the same bond that we have. So it always cleaves, chymotrypsin cleaves at the peptide bond at the carboxylic end of it. Okay, methionine, tyrosine, okay, and tryptophan. Okay. And phenylalanine. Okay. Those are clusters of hydrophobic amino acids. Okay, here's your peptide bond. There's bonds between the carbon and the nitrogen, and it's going to cleave at the carbonyl end of the carbon bond. So this is alanine, phenylalanine, and glycine put together. So remember your amino acids, and this is your bonds between here. Okay, so different enzymes are going to cleave at different places along that polypeptide chain. Okay, and this one cleaves phenylalanine at the carbonyl carbon. Okay, it cleaves hydrophobic amino acids at the carbon terminal. Okay, and phenylalanine is a hydrophobic amino acid. It's nonpolar. And it's going to cleave it at the carbon terminal. That's what chymotrypsin does. Okay, the specificity of chymotrypsin depends on the presence of a hydrophobic pocket. So you have a cluster of hydrophobic amino acids brought together by the three-dimensional folding of the protein chain. If you have that hydrophobic environment, then chymotrypsin come in there and cleave the amino acids at that junction, at the carbonyl junction of it. Okay. The flat aromatic side chains of certain amino acids like tyrosine, tryptophan, phenylalanine slide into the pocket. Binding specificity required for catalysis. Okay, so the R groups of those amino acids fit in the pocket. They fit in that pocket. It allows them to come together. It allows the chymotrypsin to cleave that and break that bond, thereby digesting the protein. So this is the specificity of the proteolytic enzyme. This is chymotrypsin. Okay, this is your catalytic. This is your active site. The R group comes in here and binds. And the R group is going to be any group that is hydrophobic. It's nonpolar. It's going to fit in there. It's going to come close proximity. So that bond can now be cleaved at the carbonyl carbon, between the carbon and the nitrogen. It's going to cleave that bond, that peptide bond. Okay. It allows it to bond to it. So it has to be, chymotrypsin affects hydrophobic nonpolar groups on amino acids. It slides into that pocket.
What does the bottom say? Oh, hydrophobic pocket. Okay, never mind.
That little thing was blocking it. Move that.
Yeah, no, the color thing is up on the bottom, so it was literally just over the word, so I couldn't see it.
Yeah, I can see. I just like to take it off sometimes. But you notice it didn't stop this time either. It didn't cut out after an hour.
What's that?
Usually my remember when I would lecture, it would cut out after an hour and have to go back, reset everything.
Uh-huh.
It's not doing it anymore. Fixed it. But it has that little thing that pops up when I draw it, but I can get around that.
Yeah, no, it's not that big of a deal because it drops down when you're not using it. So
Right. Okay, so there's your hydrophobic pocket. Okay. That's the R group that is hydrophobic that can fit in there. And then you can bond that, the peptide bond, okay, by breaking at the carboxyl end. Okay. So a pancreatic serine protease, category of enzymes, will hydrolyze peptide bonds. Okay. They appear to have risen through divergent evolution from a common ancestor. Okay, so our pancreas secretes these enzymes. They have similar primary structures and similar tertiary structures. Okay, they have different specificities as they bind to the different substrates. Each enzyme has a different pocket to fit the specificity for the side chains of their substrates. So the enzymes that are formed are going to have different pockets. These pockets are going to allow only certain amino acids to fit in there that are on the polypeptide chain. If it matches, it can fit in there, and then it can cleave that bond. Okay. Different keys fit in different locks. Same concept. Okay. So chymotrypsin cleaves peptide bonds on the carbonyl side. Okay. of aromatic amino acids and large hydrophobic amino acids. So those ones that fit for chymotrypsin. Different enzymes have a different specificity for different enzymes. Okay, trypsin. Trypsin cleaves the carbonyl side of basic amino acids. Okay, so the amino is basic, which means it has a positive charge. Okay, it can be cleaved by trypsin at the carbonyl carbon. Okay, elastase cleaves on the carbonyl sides of glycine and alanine. Those two amino acids fit into elastase, and that cleaves on the carbonyl side. Again, different enzymes are going to have different shapes of different amino acids to form from their R groups. If the R group fits in, then it can cleave that at that carbonyl carbon and separate that and form smaller peptide chains. So we can start breaking it down. So it breaks down larger chains into smaller chains. Okay, that's chymotrypsin, and that's elastase. That's what they look like. And this is the folding that allows them to have interaction with the different amino acids from the R groups on the proteins, on the large peptide chains. Okay, and you can see the different groups that can fit in. You can see how the R groups can fit in and bind in between there and then become modified and break that carbonyl bond. Okay, and that's trypsin. And there's a three-dimensional viewing of it. You see how it rotates. You can see how they can bond inside there, how the three-dimensional straight structure is held together. Okay, you see different pockets that form. And in that pocket, different molecules can be attracted, like the R groups of different molecules. So it can fit in and allow that to be cleaved. Okay. Again, your basic amino acid has a carbon group, has a nitrogen group, has a hydrogen, and then everyone's different R group. Okay. And this is the carbonyl carbon group they're cleaving right here. They're breaking the enzyme when it comes across, when it forms from this carbon. That's where we're going to break the bond apart. And there's your list of your nonpolar amino acids, the ones that have a positive charge, which are basic, the polar amino acids, so you read this thing here, okay, and the ones that have a charge, a negative charge, which are your acidic. So these enzymes will fit in different pockets. The nonpolar ones are going to fit in hydrophobic pockets. The polar ones are going to fit in polar pockets. The ones that have a basic charge are going to fit in basic pockets. The ones that have a negative charge are going to fit in acidic pockets that are formed by the different enzymes. And then you get some of the cleavage. And what that does is it allows you to start breaking down these proteins that you eat in your system, in your digestive system, the stomach, and it starts to make that chain smaller. So we can start breaking apart, smaller apart, until we get down to single amino acids. Answer a couple questions. Draw the structure formula for the following peptides and show which bonds we cleave by chymotrypsin. All right, so let's draw the, okay, alanine, phenylalanine, and alanine. You look at the, I don't expect you to memorize all the amino acids. Okay, but you look at the chart, like the chart we just had up above, alanine, phenylalanine, and alanine. Alanine has a CH3 group, okay, CH2, and then a benzene ring. So that's alanine, phenylalanine, and alanine. Those are your amino acids. That's how you draw. Remember we drew those last time? We showed you to draw these things, okay? And it's going to form a bond with the large hydrophobic group of phenylalanine. It's going to cleave that bond right there. The carbon bond of the amino acid that has a large hydrophobic group. Okay, that's where it's going to cleave. That's what's going to break apart. Okay, tyrosine, alanine, tyrosine. Tyrosine, alanine, tyrosine. Okay, it's going to cleave the bond. Again, the OH group here is going to bond into there with the benzene ring, and it's going to allow you to cleave this carbonyl bond. If there's another one over here, it would cleave to this bond here. If there's another amino acid, because that's the same group, it will fit in the same enzyme. So once it fits in, it allows you to cleave that at the carbonyl carbon. Okay. Draw the structure forms of the following peptides and show which bonds we cleave by chymotrypsin. Again, same thing. We'll do two of these and we'll call it a night. Do these two problems. tryptamine, valine, and glycine. So you look at your chart, you draw tryptophan, because you're always going to draw the same core structure right here. This core is always going to be drawn. Don't go back here. Go back up. It didn't come out right, did it? All right, so we're going to draw the core structure. We're going to include everything except for the R groups right there. All right, that's going to be the same every single time. Your nitrogen, your hydrogen. The only thing, remember, the only thing different on an amino acid is the R group. And those are the R groups. And you can get those from any of the pictures that we have showing the amino acids. Okay. So it's going to look, and the chymotrypsin cleaves had a non-polar large group to fit in a hydrophobic pocket. There's only one group that's large hydrophobic pocket, and that's this right here. And that's going to fit, it's going to cleave right at the carbonyl group. Okay. Again, you can draw any polypeptide chain, any protein. Once I give you the names, just by drawing the backbone. Okay, everything's fused by the nitrogen combined with the carbon. The nitrogen is always on the left, the carbon's always on the right when we do it, and it forms a peptide. And the only difference is your R groups. Phenylalanine, alanine, and proline. Again, so you draw your same backbone. Okay, and then you draw your R groups to it. Okay. You have an aromatic ring that's going to bond to there for chymotrypsin, and it's going to cleave between the carbonyl group and the nitrogen. Okay, so anything you'll be able to draw, and you know exactly which enzyme is going to cleave where.